Mitochondria are one of the many organelles found inside of human cells. Mitochondria are responsible for producing energy for use in the body. This energy is important because it supplies a power source to important organs in our body. For example, in cardiac muscle cells, 40% of the space within the cytoplasm, which surrounds the nucleus, is occupied by mitochondria. The specific type of energy produced by mitochondria is called adenosine triphosphate, or ATP. Shown here, cell respiration can be broken down into three steps: glycolysis, which produces 2 ATP, the Krebs cycle, which also produces 2 ATP, and the electron transport chain, which produces 32 ATP. Glycolysis occurs in the cytoplasm of the cell outside of the mitochondria. However, both the Krebs cycle and the electron transport chain depend on the mitochondria to produce life-sustaining energy. Mitochondria also have their own DNA called mtDNA, which is separate from nuclear DNA. mtDNA, or mitochondrial DNA, encodes 37 genes, which is far fewer than the 20,000 found in nuclear DNA. While small in number, these genes are still important as they play a role in the function of the mitochondria. Mutations in these genes can affect the function of the mitochondria and are sometimes life-threatening. The United Mitochondrial Disease Foundation reports that 1 in 5,000 children are born with a mitochondrial disease. However, incidence of mitochondrial DNA mutations in children could be higher because not all mutations will produce symptoms at birth. MELAS, which stands for mitochondrial encephalomyopathy, lactic acidosis, and stroke-like episodes, is just one of many diseases that can result from a mutation in mitochondrial DNA. The symptoms of this disease are muscle weakness, strokes at a young age, neurological deficits, and cognitive regression. Symptoms can also vary in severity as a result of how this disease is inherited. MELAS is passed on from mothers to their children by cytoplasmic inheritance. In this pattern of inheritance, mitochondria from the mother is divided randomly. This means that there is potential for a mother who is a carrier of a small amount of mutant mitochondria to pass on mutant mitochondria alone, which would result in an affected child. Fortunately, with research, new technology can soon be available to assist couples in having healthy children. Three-person in vitro fertilization is a solution for couples who cannot have a healthy child alone. With this medical intervention, it is possible for a mother who is a carrier for a mitochondrial disease to have an unaffected child. This procedure involves transferring the genetic material of the mother's egg with mutant mitochondria to a donor egg with healthy mitochondria. This new egg is then implanted into the mother, where the child will develop free of any mitochondrial DNA mutations. This type of germline therapy involves 3 people, 2 parents and 1 donor. What is unique about 3-person in vitro fertilization is that it grants 2 parents the ability to have a healthy child that is biologically theirs when they could not have done so otherwise. This procedure was approved in the United Kingdom in 2014, but is yet to be approved in the United States. Together, we must raise awareness for mitochondrial disease so that further research can be conducted in the United States. With this, one day, 3-person in vitro fertilization could give healthy children to affected couples across the country. This year, raise awareness for mitochondrial disease in the 3rd week of September by reaching out to educate people around you about mitochondrial disease. For more information about how you can get involved, visit www.umdf.org.